Hello everyone, the fourth lab of practical microbiology for the second stage will be about mannitol salt agar and oxidase test. So we have two procedures we will apply during this lab. Firstly, we have mannitol salt agar definition. The definition of this media. Mannitol salt agar is a media, is a selective and differential media used to isolate and identify Staphylococcus aureus from the clinical specimen. So it's a specific media for Staphylococcus species, and the color of the media is like this in figure. Why mannitol salt agar is a selective media? We have a reason. So it's a selective media because it has or it contains high salt concentration, about 7.5% of NaCl within the media, and this will only allow halophilic bacteria and halotolerant bacteria species to grow on it and inhabit other pathogenic bacteria that infect human, except we have genus Staphylococcus, the only genus that can grow on this media. So halophilic, it means salt-loving species, while halotolerant bacteria, the bacteria that can tolerate the high salt concentration. Why it's a differential media? So it's differential media because it produces acid as a result of fermentation of mannitol. Mannitol is a component in this media which is as sugar. In some species that can use mannitol, they ferment it and they produce acid as a result of these reactions or using. We have how we will know it's ferment the mannitol or that bacteria ferment mannitol. It has a pH indicator which is phenol red, where that is red orange when the pH is neutral or around pH 7. If a species ferment mannitol like Staphylococcus aureus, it will produce acid. Automatically the pH of the medium will decrease toward the acidic. So the phenol red will change its color and it will become yellow color. The principle of this media, if a particular sample specimen contains Staphylococcus aureus, it will ferment the mannitol, change the color of the media from its natural color, which is orange red color, to yellow color as a result of the acidic production from the mannitol. But if the bacteria other Staphylococcus species grow on the mannitol salt agar, they cannot ferment mannitol. Only the color of the media remain the same. Just we have yellow and does not change to the yellow. Just we have pink colony on the media will grow on it. We have interpretation of the result. When we have Staphylococcus aureus, they can grow on mannitol salt agar. They also can produce yellow colony as a mannitol fermenter, and also they can resist the high salt concentration, while other Staphylococcus species such as Staphylococcus epidermidis, they can grow on this media, but they cannot use mannitol, so they are mannitol fermentation negative, while they are salt resistance positive. Other bacterial species that cannot grow or do not grow on mannitol salt agar are both mannitol fermentation and salt resistant negative, such as we have Escherichia coli. So we have an inoculated media of mannitol salt agar. It's colored like this. When we have staph epidermidis grow on this media, they cannot use mannitol or ferment mannitol, so it's mannitol fermenter negative, and only they can grow, but they produce only pink colony on this media. When we have Staphylococcus aureus in the specimen, they are mannitol fermenters, so they change the color of the media and they can produce yellow colony on this media. Now we will start with oxidase test. Oxidase test and detect the presence of cytochrome oxidase system. that will catalyze or stimulate the operation of the electron transport chain during aerobic respiration. Well, the cytochrome oxidase system is a system or a crucial component of the respiratory chain for aerobic bacteria species, and also we have for eukaryotic cells. So it's found in the inner membrane of the mitochondria and is responsible for the last stage of the aerobic respiration, which is electron transport chain. The aim or the purpose or the use of the oxidase test, we have oxidase test is used to determine the ability of the bacteria to produce intracellular cytochrome C oxidase enzyme, such as we have Neisseria, we have Vibrio species, Campylobacter, and Pseudomonas species. They are oxidase positive. From the oxidase negative bacteria such as Enterobacteriaceae, a large family of the bacteria that have a large group of bacteria containing Salmonella, Shigella, Proteus. We have all bacteria that are oxidase positive are aerobic bacteria. They can use oxygen as a terminal electron acceptor in respiration. The principle of oxidase test, we have a reagent, which is oxidase reagent, tetramethyl p-phenylalanine diamine dihydrochloride. When we have oxidase enzyme, it will change or oxidize to indophenol. As an end product will be purple or dark blue color. When the enzyme is present, the reagent is not present or does not present, the reagent remain reduced and it will be colorless. So this equation means that when we have reagent of oxidase reagent and in the presence of cytochrome oxidase enzyme that are produced by some type of the bacteria, it will change to indophenol and change the color to purple or dark blue color. The procedure of oxidase test, we will take a filter paper soaked or saturated with the substrate or substrate with it's an oxidase reagent, which is tetramethyl p-phenylalanine diamine dihydrochloride. We will pick a colony to be tested with a wooden or platinum lobe, and we will smear it on a filter paper. Then we will observe inoculated area of filter paper for a color change to deep blue or purple within 10 to 30 seconds. And here when we have the bacteria culture, we need to examine it. We will add or smear on a saturated filter paper or oxidase test that have oxidase reagent. If we have positive result, the color of the smeared area will change to deep purple, blue, or dark blue color within 10 seconds. If it's oxidase negative, the color remains colorless. Positive reaction, the dark blue to purple coloration occurring within the 5 to 10 second only. We have example of positive reaction. We have Pseudomonas species, while negative reaction, absence of the coloration beyond 30 second. Example of the negative oxidase test, it's E. coli.